The brainstem has three parts: the midbrain, the pons, and the medulla oblongata. All of these are shown within the red circle on the slide. This is a mid-sagittal section of the brain showing the midbrain, labeled MB, the pons, and the medulla oblongata, labeled MO. All of these parts are shown within the red circle. Here you can see an inferior view of the brain with the midbrain, pons, and medulla oblongata labeled. You can also see the cranial nerves, which sprout off of these three parts of the brainstem. The cranial nerves are shown in yellow. The midbrain is the smallest part of the brainstem and connects the brainstem, pons, and medulla to the diencephalon and cerebellum. It has two cranial nerves 3 and 4, that emerge from the midbrain. And then there are a number of structures called nuclei located inside the midbrain. Those are listed on the right side of the slide. So you want to be able to associate these 5 nuclei with the midbrain. The substantia nigra is an important nucleus located in the midbrain. Substantia nigra means the black substance. And you can see in the diagram that there is black stuff on both sides of the midbrain. The green arrow is pointing to it on one side. This nucleus, the substantia nigra, produces the neurotransmitter dopamine, which is necessary for initiation of movement, emotion, and thought. And because melanin is a byproduct of dopamine synthesis, the substantia nigra looks black in a section through the brainstem. In Parkinson's disease, there is death of neurons in the substantia nigra. And so if you look at the midbrain of someone with Parkinson's, you will see that there is an absence of black stuff or melanin in the substantia nigra. That causes decreased output to motor cortex and therefore decreased output to skeletal muscles, causing the signs and symptoms of Parkinson's, which are coming up on the next few slides. This is a section through a midbrain of someone with Parkinson's, and you can see on one side of the slide that there is some black stuff left in the substantia nigra, and on the other side there is virtually no black stuff left in the substantia nigra. What this means is that the Parkinson's symptoms will be worse on one side of the body than the other. The side that has some dopamine-producing cells left will have fewer signs and symptoms than the side lacking all dopamine-producing cells. Loss of dopamine produced by neurons in the substantia nigra in the condition called Parkinson's produces slow, small movements, and the term that we use to describe those slow, small movements is bradykinesia. This slide shows the signs and symptoms of Parkinson's. The 4 at the top of the list are the most common signs and symptoms of this condition. These include bradykinesia, slow, small movement, resting tremor, which can be unilateral but is sometimes bilateral, rigidity, stiffness, and postural instability. A test for postural instability is to stand behind the person with Parkinson's and pull posteriorly. And a person with Parkinson's is usually not able to catch themselves and will fall over backwards. That's why you stand behind them in order to catch them. The masked face is due to lack of the ability to initiate contraction of the muscles of facial expression. So people look like they have blank facial expressions. Micrographia means small handwriting. Infrequent eye blink makes it look as though people are staring at you. And stooped, flexed posture. You can see that in the slide at the top of the screen on the right. You can click on this link to watch someone with Parkinson's walk. Treatment for Parkinsonism is usually to replace dopamine, and you can use that with a drug called L-Dopa, which is converted to dopamine in the brain. You can use dopamine agonists, which bind to and activate dopamine receptors, and also exercise, which has been shown to slow symptom progression. Many physical therapists now say that everyone with Parkinson's disease should be in a regular exercise program. Although Parkinson's disease is usually thought of as a motor disorder, there are non-motor signs and symptoms that accompany it, and these are listed here. Those on the left side of the screen are the most common. Notice that anosmia, or loss of sense of smell, is present in a vast majority of people with Parkinson's. Depression, anxiety, and dementia are also common. People frequently also have the things that are on the right side of the screen: constipation, bladder urgency, and fatigue, as well as sleep disorders, and those things can go together. People who have bladder urgency and frequency have to get up to urinate during the middle of the night, and so they're typically going to be tired. Pain is also possible to be found in people with Parkinson's. Another nucleus found in the midbrain is the red nucleus. It's named for its color, and the blue arrow is pointing to it on one side of the brainstem section. It is involved in coordination of movement, has a lot of connections to the cerebellum, and a small descending tract emerges from the red nucleus and connects the midbrain with flexor muscles of the upper extremity. That tract is called the rubrospinal tract. We did not study this tract, and you don't need to know about it. The superior colliculus is a midbrain nucleus that is part of a visual pathway that connects the eyes, and optic nerves, with the nerves that control eye movements. This allows us to do visual tracking. So remember the superior colliculus as part of a visual pathway. The inferior colliculus, indicated by the blue arrow on this slide, is part of an auditory pathway. It connects the inner ear and the cranial nerve that emerges from the inner ear, the vestibulocochlear nerve, with auditory areas of the brain. The last midbrain nucleus to know is the VTA, or ventral tegmental area. It is shown within the red circle in the midbrain. This is the beginning of tracts called the mesolimbic and mesocortical tracts, and dopamine is produced in the VTA. It is released in other areas of the brain during pleasurable activities, some of which are listed there on the slide: eating, sex, drinking alcohol, using drugs, and smoking. And this dopamine release is the biological basis of addiction. Every time you do a pleasurable activity, you get a little hit of dopamine. And most recreational drugs work by acting on the VTA and causing a release of dopamine. We now move on to the pons, which is shown in the top of the screen within the red circle, and is identified at the bottom of the screen by the blue and red arrow. The pons is the largest of the three sections of the midbrain. It has a lot of connections to and from the cerebellum. There are 4 cranial nerves that emerge from the pons: cranial nerves 5, 6, 7, and 8. And there are 2 major groups of nuclei to know about in the pons: the reticular formation and the ascending reticular activating system, or ARAS. And I'll describe those more in the next few slides. The reticular formation, or RF, is a complex network of neurons that's found within the pons, but that projects all over the central nervous system. It has lots of different functions. The 3 that you need to know are listed here on the slide. It controls mood and emotion, it helps to modify pain, and it is important for sleeping and waking. The ARAS, or ascending reticular activating system, is another complex neuronal network that projects to the thalamus and cortex. You can see by the blue arrows that it's sending action potentials all over the cortex. An activity of the ARAS is required in order for us to maintain consciousness. If the ARAS is damaged in some way, we will have coma. The most inferior part of the brainstem is the medulla oblongata. It's shown inside the red circle and by the blue arrow on the slides. This is the most inferior part, and it connects the rest of the brain to the spinal cord. Cranial nerves 9, 10, 11, and 12 emerge from the medulla, and there are 2 major nuclei to learn in the medulla oblongata: nuclei for respiration and a little structure called the inferior olive. In the medulla, the inspiratory nuclei stimulate the diaphragm by connections down through the medulla and cervical spinal cord to the phrenic nerve. So it's these nuclei, the inspiratory nuclei, that allow us to breathe by reflex without having to remember to do it. Don't forget that the phrenic nerve is a voluntary motor nerve, and the diaphragm is a skeletal muscle. The inferior olivary nucleus, or inferior olive, is a motor control center that's also found in the medulla oblongata. I've described for you the major nuclei found in the 3 sections of the brainstem. And the other thing to know about the brainstem is that all of the sensory and motor tracts that were in the spinal cord are also found here in the brainstem. Unfortunately, many of them changed their names in the brainstem. This slide lists the major tracts that are found in the brainstem. On the left side of the screen, there are the major sensory tracts: the lateral spinal thalamic tract and anterior spinal thalamic tract, merge in the brainstem to form a structure called the spinal lemniscus. The dorsal columns changed their name to the medial lemniscus. The word lemniscus means ribbon, and someone named them because it was thought they looked like a ribbon. The spinocerebellar tracts do not change their name. They stay as the spinocerebellar tracts. On the motor side, the lateral cortical spinal tracts are called the pyramids or pyramidal tracts when you're in the brainstem, whereas the vestibulospinal and reticulospinal tracts stay the same. I'm now going to start telling you about the cranial nerves. There are 12 pairs of cranial nerves found in the human. All of them arise from the brain. That's why they're called cranial nerves. They contain motor neurons, sensory neurons, or both. And there are 12 pairs, 2 of which are considered tracts of the brain, and 10 are true peripheral nerves that can regenerate. So cranial nerves 1 and 2 cannot regenerate if they're injured, but cranial nerves 3 through 12 are. This slide shows the cranial nerves on an inferior view of the brain. This inferior view of the brain lists all the cranial nerves so you can identify them. This slide and the next one list each cranial nerve by name and number and tell you its major function. And you also have a handout that's on the Angel site for this week that is a chart with a little bit more detail about this. For each cranial nerve, you need to know its number, its name, its function, and what would happen if there was some kind of a lesion to it. There are several rhymes or mnemonic devices for remembering the order of the cranial nerves. This is the traditional one that I learned in graduate school a long time ago, and there are other ones as well. Some of them are a little bit less than savory, so you might need to ask a student who's taken this class before to share those with you. Cranial nerves can have 1, 2, 3, or all 4 types of these nerve fibers in them. Voluntary motor to skeletal muscle, autonomic, general sensory, and special sensory neurons. Each cranial nerve is different, and so you have to learn them individually. So here I'm comparing spinal nerves to cranial nerves, all of which form part of the peripheral nervous system. As you know, all spinal nerves have voluntary motor neurons to skeletal muscle, sympathetic neurons to smooth or cardiac muscle, and sensory neurons that bring in sensation from the skin, from muscles, and joints. Cranial nerves are all different. Each one has to be learned individually. And the list there tells you the types of neurons that cranial nerves can have. Notice that sympathetic is not on the list for cranial nerves, but parasympathetic neurons can be found in cranial nerves. Also notice that special sensory nerves, which include vision, hearing, balance, taste, and smell, can be found in cranial nerves, and those are never found in spinal nerves. Cranial nerve 1 is the olfactory nerve. The arrow is pointing to it on this slide. This cranial nerve is considered a tract of the brain, and it does not regenerate if it is injured. This slide shows the upper part of the nasal cavity and the bottom of the brain, and the olfactory bulb and olfactory tract, which form cranial nerve 1, are shown. The way that this works is, when you smell something, a molecule of whatever it is that you're going to smell comes into your nose, dissolves in the mucus in your nose, and binds to receptors on the fascicles of the olfactory nerve. Those smells are then converted to action potentials, which travel through the olfactory nerve, olfactory bulb and tract, back to the brain. Here's a picture of an olfactory receptor cell that converts molecules of odor into action potentials. These are the cells that are located in the top of your nasal cavity and ultimately synapse on or connect to the olfactory nerve. Action potentials from the olfactory nerve project to or connect with the primary olfactory cortex in the temporal lobe of the brain so you can perceive the odor, and also to parts of the limbic system, and those are shown here as purple stuff on the slide. And that's so that we can have emotional responses to things that we smell. The optic nerve, cranial nerve 2, is shown on this inferior view of the brain by the blue arrow. This also is a tract of the brain, so does not regenerate if injured. Nerves in the back of the eye on the retina, which the arrow is pointing to, contain photoreceptors, and what these do is convert light waves into action potentials. So light that comes into your eye strikes the photoreceptor cells on the retina and converts that light into action potentials. We have two types of photoreceptors on our retina, rods and cones. Rods work well in dim light and they see in black and white. Cones require bright light and they allow us to have color vision. We have about 125 million rods scattered across our retina. They do work in low light and allow us to have black and white vision. We have fewer cones, only about 7 million. These allow us to have color vision, and there are three types of cones: red, blue, and green. When someone is colorblind, they are genetically predisposed to lacking one or more of these types of cones. On this slide is just a picture showing the difference between rods and cones. You can see that the rods are much larger, the cones are smaller. Once the light waves have been converted to action potentials by the retinal photoreceptors, those action potentials are traveling through the optic nerve to reach visual parts of the cerebral cortex. Vision is perceived in the primary visual cortex located in the occipital lobe, and that's shown here on this slide by the red squiggly lines. This is the site of visual perception. It's important to realize that each visual field projects to the opposite side visual cortex. This slide shows the two visual fields. The right visual field is shown in yellow, and the left visual field is shown in blue. Where those two visual fields overlap is the green region in the center. All of the light that's coming in from the right visual field is striking the retina of both eyes, right and left eyes, and then is being transmitted to the contralateral or left side of the visual cortex. Similarly, all of the action potentials coming from the blue or left visual field ultimately project to the right primary visual cortex on the opposite side. You can see on this diagram the optic nerves, the optic chiasm, which is the place where the two optic nerves cross, forming a little X, the optic tracts, which then connect back to the thalamus and from there to the visual cortex in the occipital lobe by way of axons called optic radiations. So visual information has to travel a long way from the retina in the anterior part of the brain all the way back to the occipital lobe visual cortex in the posterior part of the brain. Now we're ready to learn the cranial nerves that come from the brainstem, cranial nerves 3 through 12. Two cranial nerves emerge from the midbrain, cranial nerves 3 and 4. Cranial nerve 3, the oculomotor nerve, is shown by the blue arrow in this inferior view of the brain. The oculomotor nerve is responsible for innervating the muscles that move the eyeball, and those are shown on this slide. You do not need to learn these muscles or their function, but you can try moving your eyeball around, so hold your head still and roll your eyes up to the ceiling, roll them down to the floor, and then look from left to right, and you will be using these extraocular muscles. There are 6 small muscles that move the eyeball, and 4 of them are innervated by the oculomotor nerve. The oculomotor nerve has some other functions as well, which will be described in the upcoming slide. So, the oculomotor nerve innervates 4 of the extraocular muscles. If any of those are paralyzed or if there's damage to this nerve, the effect will be diplopia, or double vision. In addition, the oculomotor nerve also innervates a muscle that opens the eyelid called the levator palpebrae muscle. If this nerve is damaged so that the eyelid can't open, it will droop, and that's a condition called ptosis. In addition, the oculomotor nerve contains parasympathetic neurons that innervate smooth muscle in the eye. One of the smooth muscles is the pupillary constrictor muscle, and the other is a muscle that moves the lens. If the oculomotor nerve is damaged, the pupil will be dilated. In other words, it will not be able to be constricted in response to light. And you will have paralysis of accommodation, which means that the muscle that moves the lens will not be able to change the shape of the lens to let you focus up close. It's this nerve and the fact that it runs for quite a ways within the skull and is subject to pressure from swelling or bleeding, this is the reason why people will often shine a light into somebody's eyes to see if the pupils constrict. And if they don't, that's indicative of a head injury and a serious emergency. The second cranial nerve that comes from the midbrain is the trochlear nerve. That is shown on this slide by the blue arrow. The trochlear nerve innervates one extraocular muscle. Its job is to move the eyeball down and out, so inferiorly and laterally. And if the trochlear nerve is damaged, diplopia will be the result. There are 4 cranial nerves that come from the pons. Cranial nerves 5, 6, 7, and 8, and all of those are identified here on this slide. Cranial nerve 5 is the trigeminal nerve. It's the largest cranial nerve and has 3 parts. The blue arrow is pointing to the nerve. The 3 parts of the trigeminal nerve are indicated here. They are the ophthalmic, maxillary, and mandibular divisions. All 3 parts of this nerve contain sensory fibers that come from the head, forehead, eyes, nose, cheeks, palate, teeth, chin, tongue, and ear. So essentially, the trigeminal nerve is the sensory nerve from the head that conveys somatosensation from all structures of the face and head to the brain. In addition, the mandibular division of the nerve innervates the muscles of mastication, the jaw muscles that you learned about in your anatomy class, masseter, the lateral and medial pterygoids, and the temporalis muscle. The picture on this slide shows the range of sensory distribution of the 3 branches of the trigeminal nerve, V1, V2, and V3, or ophthalmic, maxillary, and mandibular divisions. So the trigeminal nerve is the major somatosensory nerve from the face and has action potentials that travel to primary somatosensory cortex for perception of touch, pressure, pain, temperature, and proprioception on the face. In addition, there are some connections between this nerve and other cranial nerves for reflexes, and an important one is the eye blink. This allows you to react to getting something in your eye and blink to clear it out. This slide shows the sensory homunculus, and I've put a blue circle around the part of the primary sensory cortex that's responsible for perception of sensation from the face. As you can see, this occupies a large area of space on our primary sensory cortex. Damage to the trigeminal nerve can cause paralysis of the jaw muscles. This is only because of damage to the V3 or mandibular division of the nerve, because that's the only part of the nerve that contains motor neurons to the muscles of mastication. You can also have loss of sensation to the face, and if one of the 3 branches of the trigeminal were to be injured, you should be able to predict by drawing a map of the face which region would be affected. The final thing that can happen with trigeminal pathology is a condition called trigeminal neuralgia. This is where pressure is put on the nerve, often from an artery that's in the head someplace. And when you touch the face, it causes an intense shooting pain that travels along one of the branches of the trigeminal nerve. Cranial nerve 6, the abducens nerve, also emerges from the pons and is identified by the blue arrow. The abducens nerve innervates one extraocular muscle called the lateral rectus, which moves the eyeball laterally. So damage to the abducens nerves will cause the eye to roll in towards the nose and will also cause diplopia or double vision. Cranial nerve 7, the facial nerve, is identified by the blue arrow on this slide. It comes right at the junction between the pons and medulla oblongata, but is usually considered to be emerging from the pons. Neurons in the facial nerve have 4 important functions. First, they innervate all of the muscles of facial expression. They also provide taste sensation from the anterior 2/3 of the tongue, and have parasympathetic fibers that innervate the lacrimal glands for tears and the salivary glands to make saliva. Here's a picture of the muscles of facial expression which you learned about in anatomy. These are very small, delicate muscles that insert into the skin and move the skin of the face in order to convey expression and emotion. These muscles lack muscle spindles, and they, among all muscles in the body, have a huge number of connections from emotional centers of the brain, so that our facial expressions can reflect what we are feeling. This slide shows the branches of the facial nerve. You do not need to know these. I've just included it so you can see how complex the nerve is. The facial nerve is responsible for conveying taste sensations from the anterior 2/3 of the tongue to the brain. The anterior 2/3 would be from the black line that's drawn across the middle of the tongue down towards the bottom of the slide. And as it says in the blue box, recent evidence suggests that this taste map may be incorrect, so it's possible that we will find this is not the true map after all. Taste sensations conveyed in the facial nerve project to a region of primary somatosensory cortex called primary gustatory or taste cortex, and that's shown by the yellow circle. Damage to the facial nerve results in the following signs and symptoms. Facial paralysis, or paralysis of muscles on one side of the face. This is a condition called Bell's palsy. I'll say more about that on the next slide. Also, because the facial nerve contains neurons that produce both tears and saliva, dry eye and dry mouth on the side of the injury are also evident in facial nerve damage. And there is loss of taste, a condition called ageusia, on the anterior 2/3 of the tongue. Here's a picture of someone with Bell's palsy. She's trying to contract the muscles of facial expression bilaterally, and you can see that on the right side of her face, nothing is happening. That is the paralyzed side. Cranial nerve 8, the vestibulocochlear nerve, is shown on this inferior view of the brain by the blue arrow. The vestibulocochlear nerve innervates both parts of the inner ear, the cochlea and the vestibular apparatus and semicircular canals, which are responsible for balance. So vestibulocochlear nerve is responsible for conveying sensations of hearing and balance to the brain. The cochlea is the part of the inner ear responsible for hearing. It's coiled up like a snail's shell. And there are small cells known as hair cells found in the cochlea. These are sensory receptors that convert sound waves into action potentials, which then travel to the brain in cranial nerve 8. Here's a picture showing what those hair cells look like. From the cochlea, action potentials travel to the brain in the vestibulocochlear nerve and form a number of synapses before finally projecting to primary auditory cortex, which is located in the temporal lobe, and that's shown here within the blue circle. You can see that there is a connection in the superior olive, as well as the inferior colliculus, as well as the thalamus. So there are a number of steps in this auditory pathway to project sound to the primary auditory cortex where it is perceived. Primary auditory cortex is shown in this slide within the red circle. The vestibular apparatus within the inner ear is the part of the inner ear responsible for balance, and it consists of two structures called the utricle and the saccule, indicated by the U and the S on the picture, as well as three semicircular ducts or semicircular canals. The utricle and saccule detect linear motion, as is shown by the gymnast here turning upside down. The semicircular canals detect rotational motion, as is shown by the gymnast spinning around in a circle. So together, the utricle and saccule and the semicircular canals provide the brain with information about the position and the movement of the head that is essential in enabling us to maintain balance. Information about the position and movement of the head is transmitted from the vestibular apparatus in the inner ear through the vestibulocochlear nerve to a number of different nuclei all across the brain. You can see here that there are many connections to the vestibular nuclei, which are found in the brainstem, and that is where the vestibulospinal tracts begin. There are also connections to the cerebellum for coordination, and also to nuclei that control eye movement, and finally to the thalamus and vestibular cortex for awareness and perception of spatial orientation and movement. So this slide summarizes the major connections for cranial nerve 8. First of all, the cochlear portion of the nerve connects to primary auditory cortex, and a pathology of this connection or of primary auditory cortex could cause deafness. From primary auditory cortex, there are several other connections that are listed here. You don't need to worry about them, but they're interesting and will come back and be important later when we learn about the cerebral cortex. The vestibular cortex has connections to other areas as well, the vestibular nuclei and cerebellum and balance centers. Connections from the vestibulocochlear nerve to the vestibular cortex and damage to those connections or to the vestibular nerve can cause a pathology known as vertigo or dizziness. Cranial nerves from the medulla oblongata are cranial nerves 9, 10, 11, and 12, and those are shown here on this slide. Cranial nerve 9, the glossopharyngeal nerve, is indicated by the blue arrow. Damage to the glossopharyngeal nerve will cause the mouth to be dry because it innervates the salivary gland. It will cause loss of taste on the posterior third of the tongue, and will also damage the gag reflex because sensation to the pharynx or throat is provided by this nerve. Cranial nerve 10, the vagus nerve, is shown by the blue arrow. The vagus nerve has a long list of functions which are summarized on this slide. It innervates muscles that you use for swallowing, muscles of the pharynx or throat, muscles of the larynx used for speaking. It also has parasympathetic fibers that go to the thorax and abdomen. These innervate all of the smooth and cardiac muscle in the thorax and abdomen. So that includes the SA node of the heart, smooth muscle of the bronchial tree, smooth muscle of most of the GI tract down as far as the descending colon and the kidney. And the functions of these connections between the vagus nerve and this cardiac or smooth muscle are listed on the slide. The word vagus comes from the Greek term for vagabond and refers to the fact that this nerve leaves the head to innervate structures into the neck, the thorax, and the abdomen. And probably its two major functions are that it slows the heart. So if your heart were not innervated by the vagus nerve, it would beat at about 100 beats per minute. The vagus nerve slows that to between 60 and 70 beats per minute for most people. It is also the major nerve responsible for digestion and peristalsis in the gut. This slide summarizes the effects of damage to the vagus nerve. Paralysis of the pharyngeal muscles could cause choking and dysphagia, which is a swallowing deficit caused by lack of coordination or paralysis of muscles of the pharynx. Paralysis of the laryngeal muscles could cause dysarthria, which means slurred or soft speech due to paralysis of vocal muscles. And bilateral damage to these muscles or to this nerve could cause death because the larynx would close over and obstruct the airway. The vagus nerve slows the heart, so damage to the vagus nerve could cause tachycardia, a rapid heartbeat, dilation of the bronchi, and could seriously interfere with peristalsis, contraction of the smooth muscle of the digestive tract, and also with urine production. Cranial nerve 11, the spinal accessory nerve, is shown by the arrow on this slide. The spinal accessory nerve contains voluntary motor neurons that innervate two muscles, the sternocleidomastoid and trapezius muscles. The hypoglossal nerve is shown by the blue arrow on this slide. The hypoglossal nerve innervates all of the muscles of the tongue, so damage to this nerve on one side will cause the tongue to be paralyzed and atrophied on that side. And you can see in this slide that this person is sticking his tongue out, and it is deviating to the affected side, and you can also see that there's significant wasting of the tongue on his left side.